Well, thanks for the invite to speak here. It's great to speak to everyone here and be at such an exciting event. I'm Frederick Bedingfield. I'm the CEO of Rubedo Life Sciences. I'm a dermatologist and a professor at UCLA, clinical instructor there where I teach and see patients in dermatology. I joined the company about a year ago. Our founder, Mark Accorda, is in the audience here. And it's really exciting time for us because we're now in the clinic with our lead drug, which we'll be telling you more about in a moment. So, one thing more just about my background. I was thinking about what some of the folks said here. So I spent a large part of my career developing drugs at Allergan, where I was the chief medical officer. Drugs some of you may know, like Botox. And so I kind of laugh now and say, I spent so much time making people look younger. Now I'm really actually trying to make them younger, so biologically younger. So that would be really quite an amazing accomplishment if we could do that. And I know everybody in the room here is kind of interested in that. So let's see if we can do that together. And it's great to have this kind of group of people all committed to this endeavor. So Rubedo, and its name is rooted in alchemy, is a private clinical stage biotech company. We're located in Sunnyvale and Milan, and we're studying aging biology with a goal of extending health span and lifespan, of course. And I think Leonardo da Vinci we'd be proud of Marco, who is an Italian especially, but all the people in this room working in this space. And we're getting older, so this has massive implications for the population. And the US Census data estimates 1 in 5 US adults will be retirement age by 2030. And as that happens, as people retire, we need other people working to continue to pay for those people who retired, and that's becoming a challenge across the world, actually. That's an expected 42% increase in age 65 plus over the next 25 years, and those 85 plus will double or triple. Caregiver numbers are falling at the same time, and chronic diseases such as Alzheimer's are really expected to double in the next 25 to 30 years. It's a real concern. And the World Bank has estimated, though, and this is the silver lining, that just 1 year increase in life expectancy can raise GDP per capita by 4% over time. And a study by Goldman in 2013 found that if you can just slow the onset of chronic disease by 2 years, you could add $7 trillion to the economy. In Rand Corporation's study, where I got my PhD, showed that workers in better health retire later and they contribute more in taxes. And so there is an opportunity here, and I would say even an economic mandate for us to be addressing these issues. So let me tell you what we're doing at Rubedo. We are in the clinic with our first drug, which is a GPX4 modulator and a senolytic. So what's the opportunity here is to target senescent cells. These are these aging pathologic cells that spew out bad schmutz into your body and cause inflammation and other problems such as fibrosis and are associated with a lot of disease. And it's interesting, there's a ton of preclinical data suggesting if you get rid of these cells, you improve a host of conditions in animal models. The challenge is that it has not really, as of yet, correlated with such a dramatic effect in human disease. And Unity, which many of you are familiar with, did some studies in the senolytic space and had some positive results, but they were mixed. And it wasn't met very well by Wall Street. And so I think one of the challenges with those first generation of senolytics is they were a bit like early chemotherapy, non-specific. And you know, today we don't treat cancer as one entity. We target a specific cell type with immune therapies and checkpoint inhibitors and all kinds of approaches such as synthetic lethality. So that's where I think senolytics have to go as well, and that's what we're trying to do at Rubedo. And we're leveraging single-cell RNA sequencing, multi-omic data, and putting it into an AI-enabled platform that we've developed to help us understand all these billion points of data, and we use primarily human tissue to derive targets. So we compare human disease tissue to non-disease tissue, AIDS tissue to non-disease human tissue, to come up with what are some of these cell types that become senescent in various diseases, what are specific vulnerabilities in those cell types that we can target in a much more targeted way, like we currently treat cancer versus in the early days. So here's our team. I've mentioned Marco, and now you've met me and the rest of the team. Most of us up in the Bay Area. I'm actually from Los Angeles, so I migrate up there frequently, but I've been here for 30 years and call LA my home. Go Dodgers. We have about 30 employees. We also have an office in Milan that we opened a year ago, and that's really exciting, and we're hoping to expand that office over the coming year. To date, we've raised about $60 million. Lead investors are Khosla, Air, Hevolution. That's the $1 billion Saudi longevity fund. CDP, this is sort of the sovereign wealth bank of Italy, has supported us. And Beiersdorf, actually, who we did a collaboration with, this is the skincare company, and they are developing, in conjunction with us, some analogs to our drugs. These are non-drugs for anti-aging skincare products. And they also invested in the company. We've also had some non-dilutive funding from the California Institute for Regenerative Medicine, and they're fully funding our second asset, which is not a GPX4 modulator, a completely different undisclosed target where we are first looking at idiopathic pulmonary fibrosis. And we're currently in the midst of a fundraising. In fact, we're closing a round right now. And fortunately, despite all these macroeconomic challenges, that's going well, and hopefully we'll have some good news to report very soon on that. So our platform, I mentioned, and we'll talk a little bit about that more in a moment, but that's how we come up with these vulnerabilities in these senescent cells. It's not a platform specific to senescence, and we are doing activities unrelated to senescent cells and other areas of aging that are interesting, and you've heard about so many different ways to approach aging here today, and that's of interest to us too. So we're not only a senescence company, but we're very interested in senescence, and we think we've got a really interesting drug in RLS 1496. And so this GPX4 modulator, we've developed a topical version, and that's in the clinic now. We are about 5 months into our phase 1 trial, which I'll tell you more about. And excitingly, it's not in healthy volunteers. Because it's a topical, we've been able to go straight into patients. So we're actually getting patient-level data in phase 1. That's one of the benefits of dermatology, and you see a lot of companies in the longevity space starting in the skin, because it's a great place to start. But in parallel, we're developing a systemic version of this GPX4 drug. And in that instance, we would be certainly looking at severe dermatologic conditions, but we'd go well beyond dermatology towards metabolic diseases, where we know this target is very important, as well as some neurodegenerative disorders as well. The second program I mentioned about, this is the one funded by the California Institute for Regenerative Medicine, is primarily today in respiratory, but that target as well has broad implications in some metabolic diseases and in some neurologic disorders as well. And actually, we just recently published some data on that in Nature Neuroscience, showing these senescent neuronal cells in the dorsal root ganglion are responsible for chronic neuropathic pain. And when you get rid of the senescent cells, you can dramatically improve that. And that's in an animal model. But we also used human tissue in that instance. So the platform, importantly, we all use these preclinical models, studying animals, and animals sometimes are good models for human disease and aging, and sometimes they're really not. But the human animal is actually a very good model for the human animal. And so we do tend to focus our efforts primarily on human tissue and human data. And so our platform really starts with clinical samples, whether it's transplanted lungs from patients with IPF that we get from Cedars-Sinai, and we compare that to normal lungs, or skin biopsies that we get from various diseases that we know are associated with senescent cells. And we put that through our platform. We do multi-omic analysis of these data. We look for, how do you, first off, even detect senescent cells? It's not as easy as you might think. And then once we've detected them, what are some of the vulnerabilities of those cells? And then we develop drugs against those targets. And then we test it again back in human ex vivo tissue to make sure we're on the right track. We put it through standard preclinical models as well, but I would say we emphasize a lot more the human tissue. So RLS 1496, in the first instance, we have this topical treatment for skin aging as well as senescence-associated inflammatory skin conditions. We're developing the oral version in parallel. It is a selective senolytic, and it's involved in a pathway that was discovered about 10 years ago called ferroptosis. This is an iron-related apoptosis or programmed cell death, and a specific vulnerability of senescent cells. And that gives some selectivity. This was discovered out of our platform in an unbiased fashion, and there are others studying GPX4, it turns out, but this is the first one to enter human trials. And there has been some interesting animal data showing that GPX4 modulation can have a longevity effect as well, as much as a 7 to 10% increase. And that's of interest to us. But in the near term, we're focused on diseases because that's a conversation we can have with the FDA today. In the long run, we hope to have a longevity drug, of course, but easier today to focus on things the FDA wants to talk about. And this is just a schematic showing where our drug RLS 1496 affects GPX4. Even a transient modulation of GPX4 in a vulnerable cell can lead to apoptosis. So you don't necessarily need a long-term inhibition, and that's what we're achieving with our drug, is a transient inhibition or modulation of GPX4. The cell that you're targeting needs to be in cell cycle arrest, and of course senescent cells are in cell cycle arrest because of P16 and CDK4/6. This drug is being studied extensively in oncology, and you may say, well, that's odd because those cells are dividing rapidly, and so how are they sensitive to this? Well, in that instance, they use a drug, palbociclib or others, that indirectly put those cells into cell cycle arrest, then they're vulnerable to the drug. And so some of the other companies that are studying GPX4 are some big pharma companies like AbbVie in collaboration with Calico, Takeda, Bayer, and mostly in oncology, some also in some chronic kidney diseases, as well as cardiovascular disease as well. But again, we're the first in the clinic, so kind of exciting, and I think there's a lot of people watching to see results from this trial that is ongoing now. We have a robust preclinical package, and we have shown out of our lab that there are a number of dermatologic conditions where senescent cells are in abundance and seem to be contributing to the disease. These are some of those listed here. Some of these are amenable to topical treatment, and some of those would be more amenable to a systemic oral treatment that we have in development. And we've got standard preclinical models and some in which we actually showed superiority to betamethasone, which I would say, in my experience in dermatology, is very hard to beat a strong, potent topical steroid in these animal models. And the fact that we were able to do that, to me, was impressive. Not as impressive as the other data that we have from humans, where I put more emphasis, though. So the study's ongoing. As I mentioned, it's a phase 1 study in patients, unusual, but you can do that with topical derm. And patients are enrolled on the basis of having really 3 conditions. So psoriasis, atopic dermatitis, and then eventually actinic keratosis in the third group. And that's occurring in the US, and we just got our US IND approved a few weeks ago. The first part 1 and 2 are occurring in Europe at a site that we're using there. And so they get this skin lesion treated, and we do pre and post assessments of that skin lesion, so the psoriatic lesion, the atopic derm lesion. But we're also testing everybody's normal but aged skin, because we're interested to see, in addition to treating these chronic inflammatory skin conditions, can we also treat aging skin and turn back the clock? and that would be very relevant to our longer-term goals, as well as our collaboration with Beiersdorf and some other activity in that realm that we have ongoing. So a lot of data coming out of this trial. I think, I've done so many trials in my life, phase 1 trials, 2, 3. This is probably the most data-packed phase 1 trial I've ever been involved in. And so we're going to learn a lot about the drug. And so between now and Q2 of next year, we'll have a ton of data coming in. So the first cohort is psoriasis, and that's your traditional SAD/MAD or single ascending dose, multiple ascending dose cohort. We're in actually the third and highest dose already in that trial. So we're well underway with the psoriasis group. We'll be enrolling the atopic dermatitis group with the high dose any day now. We'll start enrolling that. And then probably get the data by Q1, and then the data for actinic keratoses, which are, these are precancerous lesions that generally occur in older people with a lot of sun damage over the course of their life, fair-skinned people, and they get lots of these, and they can become squamous cell carcinomas, which can actually be quite damaging and deadly at times. So a really important age-related indication there that we're studying. And then we get this aging skin data as well in all subjects in all 3 of the cohorts. So what's next? So we're trying to get ready for phase 2, and this data coming out of this trial will really inform where we go for phase 2 and what we do next there. Even as such, the AK trial is almost like a phase 2A trial in essence, because we'll have already had a significant amount of human data by the time we start that. And then we're working on the GPX4 systemic. That'll be an oral drug, as I mentioned, and we're doing some modifications of that can even enhance selectivity beyond just the GPX4 selectivity of the cell cycle arrest. And there we would focus on, certainly if we see positive data in the mild to moderate dermatologic conditions that we're treating topically, we might look at treating those more severe diseases. And that's, a well-trodden path. That's what the JAK inhibitors have done with the topical and the orals, and what the PDE4 inhibitors have done with the topical and the orals, and we would certainly do that. But more exciting to us, and I say this as a dermatologist, are really the metabolic and I think some of the neurologic indications that may be possible there. And if you're really interested in extending lifespan and health span, I think those are really areas that are really going to have a huge impact in the future. And then our second compound being developed in the first instance for lung diseases and IPF, but going beyond that as well to certainly metabolic and neuro, and exciting times to come, hopefully. So this is that study I referenced. We got the cover, actually, of Nature Neuroscience, and some great work that Marco and team did with Stanford, and some exciting potential for treating neuropathic pain. We've got a great group of advisors. On the top are some of our advisors in the dermatology realm. These are very much key opinion leaders and ex-presidents of the American Academy of Dermatology. And then on the bottom, really our scientific advisory board from various walks of life and disciplines advising us, and we appreciate their support very much. And I thank you for your time, and I'm happy to take questions if there's time.
One very, very quick question. Anyone? No? Oh, Suzanne. Greg, why don't you?
Your RBX002, and you talked about fibrosis. There's a situation of bladder fibrosis after men have had radiated prostate cancer.
Sure.
And they're using Botox now. Do you see that this is a possibility?
I think it'd be very interesting, and one of the ways in which you induce senescence is through either radiation or chemotherapy. So I think, this is a big, it goes, beyond bladder cancer. I think the post-cancer treated patients, whether it was with radiation therapy, which clearly is an enormous way of introducing fibrosis and senescence, is an opportunity, and also chemotherapy as well. So those are really quite of interest to us, and we'll hopefully be able to do some work in that soon. We're not currently doing that work, but we're very interested in it.